thanks everybody for showing up for this talk. I know it's very late in Europe, and I don't know if there's anybody from who tuning in very, very early from Australia. So this is a workshop that Avi, Srivastava, and I have put together. So you've got two developers of this workshop today, and it's aimed at people who maybe are somewhat familiar with single cell already, but just they have a concept of the idea of the kind of count tables that are generated in single cell data, but we don't assume very much in terms of method knowledge beyond that. And so I think we'll go pretty slow today talking about, so first I'm gonna hand off to Avi, who developed this Alevin software package. It's not a Bioconductor package, it's distributed part of Salmon. And Avi will talk a little bit about what goes on when you run Alevin to quantify single cell data. And then I'll talk a little bit about importing it into Bioconductor, in particular using this tximeta package. And I wanted to mention a couple things. Let me just pull up my reminders, things I wanted to mention. So here. So yeah, please post your questions in the poll on Pathable. That would be great, so we can kind of see your questions as they come up. And also, I definitely wanted to mention Charlotte Sonneson tomorrow at 3 p.m. is going to give a talk. So it's in the contributed talks number 6 session, which is related to Alevin and tximeta. So she's going to talk about some work in doing velocity quantification. So if you're interested in this, you'll probably also be interested in that. So I will, I'm going to pull up the tutorial for today, and I'll be driving. Let's see, I guess I'll pull up, I'll start from here and then I'll switch over to the Docker. So Avi, did you want to take over talking about the introduction to Alevin?
Yes. Okay. So thanks, Mike, for handing over and letting me be the part of the workshop. And thanks everyone for joining. So basically, before we move on to a very magical R word, I just want to give some motivation. Why do you want to rethink about how you actually get the gene versus cell count matrices, which lots of R packages are written around and has been processed to do cool visualization or generate downstream analysis from them. So last couple of, let's say 4 or 5, half a decade, or from 2015, 2016, this single cell word has been really blown away the number of sequencing, number of cells which you can actually sequence. And the high throughput sequencing can generate initially thousands of cells, but now you can assume we can have millions of cells in a single experiment. So obviously, if you have million cells and typically 50 to 60,000 gene or transcripts, you need faster methods so that you can analyze them within a couple of hours and iterate upon based on what chemistry you want to change, or let's say generate meaningful results from the counts. So we designed this Alevin framework from 2019. There are lots of competitors around this, especially CellRanger, Bus Tools, and Star Solo Pipeline. Bunch of them are still in still getting, let's say, building up and maybe peer reviewed, but Alevin has been still published and it's been verified through peer review process. So what specifically Alevin brings? So if you compare about CellRanger, right? So specifically what CellRanger does, it's when it starts to generate the gene versus cell count matrices, it looks for reads which multi-map across genes. And if it multi-maps, then it just throws away. And that's basically every other pipeline out there does. So what if you can use them in your principal frame, use those multi-mapping read in a principal framework and generate a gene versus cell count matrices by basically not throwing away a quarter of your data set. So if you try to do this analysis, how many multi-mapping reads you are actually tossing away before doing an analysis downstream, sometimes can be 25% of the data set, or basically varies between 15 to 25%. So a major fraction of the data set is just getting thrown. Moreover, it's not just how you throw away the data, it's a bias which you are throwing away the data specifically from the genes which have highly ambiguous region. So you can imagine like pseudogenes or genes which actually shares a lot of, let's say, their genotypic sequences with some other genes, will just throw them away and you will observe no count to them. So this bias is actually can be reflected downstream if your gene of interest is from these specific kind of situation where where a read can multimap and pipelines are just throwing it away. So with this motivation, Alevin was designed to model in a principal framework with one more thing in mind, that everything is one streamlined process. So before Alevin, things were like you combine multiple tools and then generate the gene count matrices. With Alevin, you just give Alevin the FASTQ files, and it'll go through multiple processes like cellular barcode selection, UMI deduplication, and whitelisting of cells with the mapping of reads to the reference, and it'll give you cell versus gene count metrics. So it's a one streamlined process, which makes it very useful and less stressful when you're trying to analyze your data set. So when the natural question comes up, like how do you use Alevin? So before we start using Alevin, we have to actually reference the FASTA file from using Salmon index command. I'm not going to go into much detail because if you're coming from bulk RNA-seq word, you know how, I'm assuming you might know what is Salmon indexing. But just to give you a brief overview, what it takes is the referent transcriptome and saves it into a disk so that if you map a read onto the reference transcriptome, it can be mapped at very fast pace, and you can generate the results. So that's the basic format of, that's the basic idea of indexing the reference. So once you have the index of the reference, as you can see in the running Alevin pipeline, this is the general framework of the command which you have to run as Alevin as. So if Mike, can you just, yeah, so wherever Mike's arrow is at, so there is a cool GIF at the black, shown at the black at the bottom of the screen as well. But just to give a brief overview, what these flags are, how can you tweak these flags and can generate results what you wanted to, basically count versus gene versus cell count matrices. So basically, you start with Salmon, which is once you have your Salmon installed through Conda or through a Linux binary, it starts with a, let's say, head command, which is Salmon, and then there's a sub command, which is Alevin. And first thing which you have to specify is the library type. This is important. Why? Is here, if you can see, it's ISR. What does ISR mean? ISR means what is the expected library type when you actually sequencing the data set. So the basic idea was Alevin was designed was over the droplet-based sequencing, specifically drop-seq data, in-drop data, or 10x chromium 3' seq data set. So in 10x 3' or droplet-based data sets, where what you expect, one end of the read will have cellular barcode and the UMI, while the other end of the read will have the actual sequences which has to be mapped, right? And usually what happens, one end is in the reverse strand, and that's why we have to specify ISR with. Basically, it means that reads are facing inwards and they are stranded, and the one which has the read components in the reverse strand, right? You can assume that their 10x 5' sequencing have come along as well. So what you do to what you have to change when you're providing 5' sequencing, just change ISR to ISF. Basically, you say that the reads are coming from the forward strand since it's a 5' sequencing. So this is a basic change in terms of what it does. It helps Alevin to map selectively the reads which are coming from one strand and actually give lower probability for the reads which are coming from the opposite strand, since we expect less those kind of reads, right? So this was the basic strategy for why we need a library type, which is shown with -L command. Now, the second thing is -1 and -2. Basically, what it tells that -1 file would be the first file where we expect a cellular barcode and the UMI sequence to be present. And -2 would be the second file where you actually provide the read sequences file in general. In general, these two files are generated with dropseq, indrop, and 10x sequencing, but you can imagine sequencing methods like newer methods are coming, let's SkySeq and all those combinatorial indexing data set, right? Where what happens is people just divide these two files into three files, which are, one would have cellular barcode, the second would have the UMIs. Basically, in that scenario, what we are going for now is to process them before even running this Alevin pipeline. You have to process them and concatenate them into one file and provide into this -1 command if the cellular barcode and the UMI are in different files. So there's one very exciting development. A new total Rust-based pipeline for Alevin is coming out, which will handle these kind of situations. If you have cellular barcode and the UMI information in 3 files or 5 files, it can handle very beautifully. But it's still in the pipeline. But hopefully it'll be out very soon. So these are the two -1 and -2 flag. And if you go further, you can see the Chromium v3. So Chromium v3 is the meta flag here. So what is a meta flag? What do I mean by meta flag? What it basically does is it sets a subgroup of internal flags, right? You have to tell Alevin what is the length of the cellular barcode, what is the length of the cellular UMI, and which end Alevin should extract it from, right? So there are 3 flags: UMI length, barcode length, and end. So if you don't specify them explicitly, you can specify these meta flags. We have predefined them based on what we expect in a sequencing technology. So for Chromium v3, generally cellular barcode is 16, then UMI length is, let's say, 12. So it internally sets it if you specify Chromium v3. If you just specify Chromium, it'll just set cellular barcode 16 and the UMI length 10, right? So I just want to be explicit. People talk a lot about that can Alevin be tweaked to make sure that a new, let's say, custom cellular barcode length can be provided. So yes, Alevin can be provided with custom cellular barcode and the UMI length. You have to just provide minus UMI length with the what is the expected UMI length, and minus barcode length with what is the expected barcode length. And you do not have to give these meta flags. But basically what meta flag does is set internally these barcode length and the UMI length flags. Now, if we move further, there is another command which is index. As I was talking before, we have to run Salmon index, and this is basically the location or the path in the disk where you have run Salmon index on, and it stores the index to map the set of reads to a set of reference which was used in the Salmon index command. Again, Alevin is very fast end-to-end pipeline. It can use multi-threading very efficiently. -P basically tells Alevin to how many threads to use when you are quantifying your data set. If we go further, there's -O, which tells where to store the data set, where to store the output matrices, basically the cell versus gene count matrix after you have done the quantification. So, and the last one is this tgmap command. Basically, you have to give Alevin file which gives you the relation between what are the set of transcripts and what are the set of genes which they come from, so that Alevin can perform these grouping based on transcript level and then they perform UMI deduplication to generate the gene cell versus gene count matrices. So one very important flag which we are very excited about, especially Mike have put a lot of efforts into it, is this minus number of cell bootstrap. So let me give you a little backstory before I try to explain what it does. Is as I was talking that what Alevin does is it tries to take the reads which is multi-mapping across a bunch of genes, right? What it does is it's use expectation maximization algorithm to distribute fractionally one read across the genes where it gets multi-mapped, right? Now, it uses some likelihood estimation to optimize the overall global estimates so that it can be distributed across the genes which a read is actually multi-mapping to. But you can imagine if you change the sampling distribution a bit by a smaller margin, then the distribution, how the read is getting distributed across multi-mapping gene can change, right? And as if you keep changing your distribution, expected distribution of likelihood, you will get different distribution with each new round of bootstrapping, or how you change the function of distribution, right? So what this flag number of cell bootstrap does, it's changed the initial expected distribution of multi-mapping of the sampling across the multi-mapping genes, and it gets a new estimate through bootstrapping. And basically what this number of cell bootstrap, a number which is shown here by 30, it tells Alevin that you have to perform 30 rounds of these bootstrapping. So what basically it does, it actually blows the whole space of cell versus gene count matrix. So you can intuitively imagine, if you imagine this 2D, like one dimension is cell, another dimension is the gene, you can now assume a third dimension, which is a cube, which you're taking a square and converting it to a cube. This matrix becomes 3-dimension, which is one dimension is cell, number of cells, second dimension is number of genes, and the third dimension is how the estimate change across multiple bootstrap distribution. So what it gives you a confidence, what it can give you a confidence estimates across each cell versus gene count matrix. And you can use that, maybe Mike is going to talk into more detail, how you can use that, this cube instead of one square, and increase your downstream analysis much for the confidence in your downstream analysis, much more in terms of their efficiency. So this is a very short version of the GIF where Alevin starts to process a very small data set. I would kind of suggest to look into it, but the whole thing is online, but the data set which I am showing here it's very small data set. It's almost like hundreds, I think 1700 cells, but Alevin was able to quantify them relatively very, very fast on the mouse reference. It took like 14 minutes to quantify like 33 million from end to end, starting from FastQ, and generate cell versus gene count matrix with 30 bootstraps. And which is very important, you were able to generate a cube of the whole thing. Like you were able to generate 30 times the cell versus gene count matrices within like 14 minutes across 1700 cells. So that was just a gist of what it will be able to do and how to run Alevin. So just to, if you go a bit further, Alevin QC. So again, Charlotte has designed a very awesome method in R. What it does, it takes Alevin output directory and it gives you cool plots again on basically to perform a quality check on how your output was generated, and it's called Alevin QC. And if you can go to this website from Charlotte, it generates very cool plots. Once you specify which specific directory the output of Alevin is stored, and it generates a bunch of plots, like specifically on the top left, you can see how the cellular barcode and the frequency of the initial reads were coming out, and how Alevin is selecting accounts among what are the high confidence cells, among them which you are sub-selecting across the cells which are present from 1700 cells. On the left bottom, you can also see that she was comparing number of what is the UMI deduplicated counts across cells. On the x-axis is the number of barcodes. On the y-axis is the deduplicated UMI, which is expected a linear relation here, which I think is because of 10 times the UMI deduplication. But you can see how we can use as a quality check measure for output of Alevin. With this, I would like to transfer back it to Mike.
Great, thank you.
Yep.
One thing I wanted to mention before I keep going, if you go to the chat on Pathable, so you can put questions into the polls tab. In the chat tab, I put a link to the, I guess if you've been to these workshops before, you know where to go to create an image hosted by cancerdataSci.org. But you can click that link and pull up our workshop today. So if you wanted to follow along, Sean sent me a link where I can see in the back end that there's, I think, something like 12 people who have an instance running right now of our workshop. But if you wanted to do that, you can try that out. Okay, so I'm gonna switch over from the the website vignette to our studio instance. And I've got the vignette on the side here. Let me maybe resize a little bit. Okay, so Avi was mentioning that we're interested in the uncertainty from the assignment of reads with Alevin. So we perform EM to keep all of the mappable reads and not discard those which are gene multi-mapping, and we use bootstrapping to get an idea of the uncertainty of those assignments. And conceptually, it's a cube. But what we found recently is that we can efficiently compress that to just the mean and the variance of the bootstrap estimates. And so there is an option in Alevin to store the entire cube except not the zeros. So in the output of Alevin that we read into Bioconductor, we don't store the zeros. We only store the non-zero counts and their locations. You can also, so you can store the entire cube, so the sparse cube, but we've found, and in this workshop, we only have Alevin outputting the bootstrap mean and the bootstrap variance, and we found that that's sufficient to then make use of the uncertainty in the assignment. I'll start over here. We're going to use tximeta to import the Alevin counts into Bioconductor, and let me see. I should also pull up the vignette so I can be running code from the chunks. Okay, so I'll put. Okay, so let me just scroll down to our relevant spot. Okay, so this is this is non-typical code. This is just because we have our data is actually stored in this package. So usually you would just specify dir being where the output data from your Alevin run is. In this case, it's inside of this Alevin to BioC package. So that's not typical. We will just make sure that the files exist where we think they do. Sorry, this is going to bug me. I forgot to, on this instance, not have the output be shown in the R markdown. So give me 1 second to turn that off. Okay. And then so let me talk a little bit about this line here. So the line to read in the quantifications from Alevin into Bioconductor is a single line of code here. So we load the tximeta package. tximeta is a package which builds upon tximport. So if you've worked in RNA-seq, you might have heard of tximport, which is a package I developed with Charlotte Sonneson and Mark Robinson's group to import quantification data into Bioconductor. Kind of works across different quantification methods, and then allows you to use that quantification data with various different downstream packages. So the goal there was modularity. tximeta builds upon tximport, and instead of just having a list of counts and abundances and links, we build a summarized experiment. And so the additional thing that goes on in tximeta, which let me start this code running. So here we just point it to the files is just a single, a pointer to a single quantification matrix on our machine. And what goes on in addition to what would happen with tximport is that tximeta recognizes what genes were used for quantification. So the first time that you run tximeta on your machine, it wants to know where should it set a cache, because tximeta will be downloading annotation data based on what it recognizes the genes that you use for quantification. So it basically asks if it should use a default location or not. And so in this case, we're going to say, yes, we want to use the default location. You could say no, and then specify where you want the cache to be later. And then it asks again, can I create this, this cache directory? So it's just using kind of a standard location for the cache. And the next thing it says is that it found, looking at the quantification data, it recognized that we're using GenCode for Homo sapiens, and we're using release 33. And the way that it did this, like the magic behind that recognition, was that Salmon and Alevin hash, create a checksum basically of the transcript sequences during quantification. And that checksum can then be used for reverse lookup of what genes and what basically the provenance of the genes or transcripts that were used for quantification. So right now this works, tximeta works for bulk data or for this Alevin single cell quantification for human, mouse, or fruit fly, but we hope to very soon extend that out to actually all organisms that are from Ensemble. And so GenCode is just, we fully support GenCode now, but we're going to expand out to other organisms. So what tximeta does after it recognizes which transcriptome was used, it then automatically downloads the transcriptome if it does not exist in Annotation Hub. So if we used Ensemble, that would already exist as part of Annotation Hub, and it would then pull down the Ensemble DB database. In this case, the GenCode transcripts and genes do not exist in Annotation Hub, so it needs to download and parse this file. And a couple notes there is that this download only happens once. So, because we're using BioC file cache, if we were to quantify with this and import data again, this particular transcript set, it would not re-download or re-parse those files. It's stored in the cache. And let's see, I seem to be kind of stuck right here. I might stop this and try, well, okay, there it goes. So there's just a little lag in downloading that GTF file. Another thing I want to point out is you could use the same cache location for multiple users. So if you had a group on a Linux cluster, you could all specify the same cache location, and then it wouldn't be downloading and parsing these files if someone else in your group had already imported data from GenCode release 33. So we kind of try to make that as efficient as possible to avoid this kind of download and parsing. I think maybe I'll just switch over to the pre-cooked cake on the right-hand side while this thing is parsing. So what's going to happen next? So right now it's creating a gRanges object for the genes to attach as the rows of the summarized experiment. Yeah, for some reason it's kind of laggy right now, but this is on my machine. I don't know why. So I'll just move along on the right-hand side until it catches up. So what we get back from the tximeta call, like I said, is a summarized experiment. So SE, but if you've done work in Bioconductor, you know that there's this single cell experiment which kind of builds on top of the summarized experiment, and we probably are more interested in working with a single cell experiment. So it's very easy to just convert with the as call from our summarized experiment to a single cell experiment and then proceed with this SCE. So, you can take this object SCE and then go along, for example, with the Orchestrating Single Cell Analysis book, this really great online resource here. So this is kind of strange. On the left-hand side, it's going really slow, making a TXDB object. That's usually not that slow. Maybe it's something to do with the, I don't know, because I'm also running Zoom or something. So I'll just continue on the right-hand side. So you can look at, we have this summarized experiment or single cell experiment. You can look at the assay name. So it has 3 assays. It has the counts, which are the MLE, maximum likelihood estimates of the counts from Alevin. We also have the bootstrap variance and the bootstrap mean. So these are kind of extra information that we get specifically with Alevin. And like, I think many of you are probably already familiar how to access the assays of a summarized experiment. You can use, there's various different ways but we can use, for example, the double square bracket indexing. And this is just to point out actually that our counts here are sparse. So we've saved this using we've read it in it's saved in a sparse format from Alevin, and it's also read in a sparse manner. So we kind of avoid ever reading in these zero counts, and we only save the non-zero. Yeah, so we can also, because this assay is called counts, we could also in this case use the count successor. And I guess I have a little demonstration of like what are, aside from having the genomic ranges automatically attached programmatically to our single cell experiment, other benefits of using tximeta, because it knows the organism and it knows the type of identifiers on the rows, we can easily do things like add alternative gene identifiers. So you can add the symbol, for example, and now we return a single cell experiment where the symbol has been added in the case that there is a symbol to match with. So then, this is useful because if we wanted to, for example, compare with other single cell experiments, a lot of the single cell experiments on Bioconductor or elsewhere are have symbols as the gene identifiers. Another benefit is that we already have ranges attached to the rows. So for example, if we wanted to zoom in on a particular region, say like 100 kb region on chromosome 1, we could just use the single square bracket indexing to pull out the subset, in this case 4 genes that fall within this 100 kb region. So I'm gonna switch back to Avi so Avi did a little bit of R scripting. Out of his comfort zone maybe. So Avi did the adding of cell annotations for here. So maybe I'll switch back to Avi who can explain this part.
Yeah, so what I did basically was once you have the cell versus gene count matrix, you can have a summarized experiment or single cell object. Also, since it's just a matrix, you can create a Seurat object. So basically these are two different words. You can use the Alevin counts to analyze in either way. And what I did is basically to get the annotation, take the cell versus gene count matrices, imported them into Seurat objects, and try to make a reduced dimensional representation using PCA, and extract what are the specific and cluster them using the Seurat's algorithm. And for each cluster, identify what are the marker genes, or not marker gene, what are the differentiated genes across each cluster, which are different from, which are differentiating a specific subgroup of genes, either as differentially expressed higher or lower in specific subgroup of clusters. And once we know what specific subgroup of genes are differentially expressed, we can use let's say marker gene information to assign each cluster annotation based on what specific subtype of what cell type cluster can be assigned. And I use some of the marker gene information. I think it should be in the, if not, it's right there. I just assigned a subgroup of cells which are clustered together as one sub cell type. That is obviously needed for the downstream analysis, which Mike is going to talk about.
Okay, great. And I'm in the meantime, I'm going to switch. So I don't know what happened to my my local version. So I'm going to switch to, can everybody, do you see the new version, the new, okay. So I'm switching over to cancer data side because I think something went wrong with my local version. So just bear with me. This is how it always goes, right? I'll just run up until, yeah, let me rerun this while we talk. Okay, so I'll rerun up to tximeta. So, and I have to load the vignette on this side. Okay, so, oh, and I know one more thing, sorry. I have to do the same thing again to ask it not to put the output into our markdown. Okay. The good thing is that it's realistic. So if I were to do this on my machine after having run this before, you wouldn't see the prompt that you should ask where the cache should be. So you're getting exactly the same thing that you're getting on your end if you're running these this workshop with us. Okay, so we have this single cell experiment. We have counts and bootstrap mean and variance. And now we've added these cell annotations, which we used the separate script that we provide with the package. Yeah, see that's, by the way, this is more typical speed, that it would go pretty fast in the making of the transcript database. So I want to point out, once we have mapped cell annotations, one thing to keep in mind is that, and that's important to consider when you're doing analysis, is that the different cell types, or let's just say clusters, have different total counts. And that's something that you want to take into consideration when you're doing scaling or normalization. So in this case, if we make a very simplistic binary division of the cells, those that have more than 10,000 mapped UMIs, then we see that it's this distinction, those cells that have more UMIs is really imbalanced across the different cell types. So it's just to point out that you want to take this into account, and that's down below. When we do some scaling, we'll actually demonstrate that we use a method from Scran, which does take into account that there are kind of clusters of the cells when it's performing the scaling normalization. Okay, yeah, so this is more typical speed on the left-hand side. Let me just run up until where we are. Okay, so just in case we want to use the live stuff, so I'm going to run there. So we kind of, in the very beginning, we mentioned that that there could be many reads or data which is lost when we throw away the multi-mapping reads. And just as a visual of how much is lost, we downloaded a recent data set of mouse embryos. So this was just published last year. And just to give you kind of an idea in your mind, when you think about if you do not use EM for generating single cell count data, that's on the left-hand side. So each point here is a gene where I've summed up the counts across all the cells. And then on the right-hand side is the same gene quantified, preserving the multi-mapping reads. And I'm not showing, in this case, I'm not showing the genes where the counts are similar. So I'm only showing the genes where there's a discrepancy between the two methods. As would make sense, the counts with the EM are always larger because the no EM method of quantification is the one that's discarding data. So it can be quite substantial. But when we do this, so when we perform EM, we could be assigning these reads, but have a high uncertainty of their of which gene they assign to. And so when we make use of the extra data, we also have to bring along information about that uncertainty. And so we have we've made some plots, some plotting functions in a kind of a tag-along package called FishPond. So FishPond is basically a set of a bioconductor package which kind of assists with using data from salmon or from Alevin. And we've made a function that allows you to kind of visualize the uncertainty per cell, because the uncertainty is posterior, sorry, the bootstrap variance is attached to each gene and each cell, you can visualize that uncertainty for a given gene across cells. So let me see. I'll do this in the in the session actually. That's strange. It ran that again. Anyway, so it ran that again, and that was very fast the second time because it was already cached. So we can generate this plot. So here, each point on the x-axis is a cell. On the y-axis, we have our counts, and we're showing the bootstrap mean with the kind of solid dot, and then the band indicates the bootstrap variance on that estimate. And if you have groupings, in this case, like we do, you can specify a grouping, and then it'll kind of order the cells within the grouping. So yeah, one thing I want to point out, before I ran this plot infreps command, I ordered the cells by their column sum, and that's not a necessary or typical step. I really just did that for pedagogical purposes so I could show you what it looks like before and after scaling. I'm looking at the time, so I know that I probably won't be able to get through. I don't intend to, get through all of the workshop today. The point is that just to give you a taste, and you can kind of look at it on your own. But I do want to show you one thing, which is the scaling, and then I'll also jump to the end to show you kind of taking this data set out and then using it with Seurat, for example. So yeah, I might skip this. This plot that we created adapts to the number of cells that you have. So kind of, if you have less cells, we kind of increase the resolution. So now we're looking at only 100 cells, and we kind of increase the features of the plot as the number of cells increases or decreases. So if we don't reorder, so these plots are ordering the cells based on the bootstrap mean. If we don't reorder the cells, you can tell that the total count is obviously a technical issue here, right? Because the first cell in each group is typically has like the largest count. So that's something that we would need to take into account. And then we have some chunks here where we show using the compute sum factors from the Scrna package to normalize. And that should be pretty fast. And then we can use this apply size factors argument to, so this top plot here is not applying size factors, and the bottom plot is basically dividing out the size factor before we make the count. So on the top is counts, and on the bottom is the scaled counts. And let me see, I think I'm gonna, I might switch over to the rendered vignette just for the very last pieces. So basically we showed plotting the count and the variance, the bootstrap variance, and we can look globally at like the distribution of the variance, and we can see that generally there's some kind of not too much multi-mapping and uncertainty, but then there is a clump with a lot higher variance. And these are kind of the genes where it's important to have that bootstrap variance information. And I'm gonna, we kind of dive in deeper into what genes those are. And then the very last thing I wanna show you is, for example, if you wanted to, so Avi is in the group that develops Seurat right now as a postdoc at New York Genome Center. So Rahul Satij's group. And it's very easy if you wanted to go off and use a Seurat workflow to convert this single cell experiment object into a Seurat object, and then, for example, make some Seurat plots. So I think I'll stop there and see if there's any questions.
So there seem to be quite a few questions in the poll, so I can start giving you guys some questions if you'd like. So the most upvoted question is actually for Avi. How long till you convince Rahul to on adopting single cell experiment for Seurat.
And there's like a winky face too on there.
So to be honest, I was not involved in either of the process. So I can't say much about that. That would be my answer. But I would say they both have their own pros and cons. So I think I'll let the leaders of the projects to decide among themselves about that.
Sounds good. Just gained 2 votes in the meantime, so it's up to 10. What is the approach to using Alevin with single nucleide data? With CellRanger, it's recommended to use pre-mRNA reference by using a GTF, where feature type transcript is extracted and replaced by exon. Any particular recommendation for Alevin-Salmon index process?
Yes, that's a very good question. And again, Charlotte's name keep coming up. She's doing awesome job in almost every project she does. So she recently, she actually talked today, and she's going to talk a lot about how you can use intronic sequence or specific subgroup of sequence to actually index with Alevin to actually post-process them after quantification as an RNA to generate RNA velocity plots. For single nuclei, I am still not very much convinced that what would be the way, let's say, recommended way to go with respect to include the to include the intronic sequences or not. In general, I have found inclusion and non-inclusion of the, let's say, pre-mRNA or not immature mRNA doesn't make lots of difference in Alevin quantification, but my sample of experiment which I tested on was relatively very low, and this one on the open 10x dataset. So I would recommend going through Charlotte's tutorial and maybe do some experiments based on that and index the intronic sequence and try to see what, let's say, inclusion into the Alevin's indexing changes the results of the quantification.
Yeah, just a plug for, I mean, I assume Charlotte might talk about this tomorrow at the talk session, but there's a new function in tximeta for splitting a summarized experiment into, for example, like exonic and intronic. And she's written this split SE function here.
Great. Next question is the expected value of the bootstrapped quant values equal to the estimated gene expression count based on the full data?
There are cases where the mean of the bootstraps is not close to the EM, so the MLE, and those are very interesting for us. Those are cases where we tend to think that the bootstrap estimates are giving us some important information. So that depending on the actual method of the EM, or ways that we implement the EM, we can end up with kind of spurious zeros there. And yeah, so that usually, actually, I would say usually they're the same, but there are cases where they're not. And I think it's important to take into account the bootstrap mean and variance.
How do you handle multiple samples? Do each need to be read in separately with tximeta and then somehow combined?
Yeah. Right now, this thing is just giving you a single, one single cell experiment, which is one sample. Avi, do you have thoughts on processing of multiple samples?
So like I was saying, when Alevin was designed, it was designed for, let's say, 20 or let's say 100,000 cells. Right now, we are at like million cell in one experiment, specifically if we talk about combinatorial indexing. So we are scaling things up by quite a lot. And like I was saying, there's a Rust implementation coming very, very soon. And we're going to modify a bit about TX import if we see the use, like if it's taking too much time. But that's quite a bit of experiment. But for 100,000 cell, it should not matter much. But if you talk about million cell, we are in the experimental stage, even for quantification and then for the import downstream.
Oh, sorry, that wasn't for multiple or million cells, as it was if you have multiple libraries.
Sorry?
So if you have multiple libraries from different samples.
Yeah.
And not just one.
But usually what maybe I'm wrong, but they are quantified together because of combinatorial indexing, no?
Not always. Are the ones we have aren't. They're separate. We get separate FASTQ files for each sample.
Okay, I thought the dyscRNA-seq, you have multiple round of barcoding and everything, even though his library is separate, you have to quantify them together. But maybe I'm not sure. I still have to experiment on that.
Okay, well, they keep changing what they're doing, so it's always a game of trying to play keep up. Thank you.
Yep.
All right. Is there a way to keep transcript level count during tximeta?
Yes. Well, so I'll comment and see if Avi has an additional comment. So if you have full length single cell like SmartSeq, in that case we're just using salmon, and so yes, you would import it. You could again import it with tximeta, but you would use type equals salmon. Does that sound right, Avi?
Yes, that's a very important point. And I would argue in 3', like I totally agree with Mike, if you have say full length protocol, you can. But in single cell 3' sequencing, I would argue there is not much of a signal itself to generate, let's say, transcript level sequencing, or let's say allele level counts per cell. And it would be really noisy to have that. And we might need better methods downstream to actually process them, even in 3'. Uncertainty would be too high at isoform level to generate the count, especially in 3' sequencing.
And there's another, I see there's another question below related whether we do length offset, and we do not when we do 3' tag because we don't expect a length bias. Yeah. So for 3' tagged, we're immediately summing to the gene level and not using a length offset. And then for full length protocols, we're just using salmon, and then we are having a length offset.
Right. Probably time for just one more. Are the multi-mapping reads taken care of as RSCM does in bulk RNA-seq? What is the optimum bootstrap value?
I guess there are two different questions, right? So the first one, I guess, is saying, is multi-mapping read somehow related to TPMs in the bulk RNA sequencing modified, understanding correctly? I would say here 3' sequencing is actually an absolute count. It's not a relative count. In bulk RNA sequencing, it's actually a relative count where you normalize everything to 1 million and use length bias and positional bias. But in 3' sequencing, there is no correction for length bias, and it's absolute count in a pseudo sense because you are actually taking one read and dividing into a fraction based on the distribution. But overall, if you sum the number of counts, it's not going to sum up to million. It's going to sum up to number of reads. Number of deduplicate UMIs, which are supposed to be distributed. But I do not understand the second part of the question.
I think I get it, or at least I'm going to use it to promote this preprint. So if you're asking how many bootstraps to generate, we have a paper with the Rob Patro's group, including Avi and people from my group at UNC, this Van Buren 2020, where we look at the coverage if we simulate a single cell data set and then perform Alevin with the bootstraps, looking at bootstrap interval coverage of the true value. And we found that I think it was 20 gave the same coverage and good coverage as opposed to like 100 bootstraps. So it seems like 20 is sufficient. And also just storing the mean and variance was sufficient to get the same coverage. So that's this, it's just went up in July, this preprint down here.
All right, so I think that's time. There were like 3 questions left. So you can always copy the poll questions and send them in the chat. The chat sticks around for a little while.
Yep, I copied them.
Perfect.
Thanks everyone for attending this session. I know it's probably not perfect convenient time. And thanks Avi for helping out and Kayla for moderating.
Thanks, Mike. Thank everyone.